The impact of folate on epigenetics offers a fascinating glimpse into how nutrition can transcend individual health and quietly shape biological destiny across generations. Folate, long respected for its role in supporting fetal development and preventing neural tube defects, has a deeper and more subtle function as a regulator of gene expression through epigenetic programming. This involves chemical modifications, particularly DNA methylation, that determine how genes are expressed without altering the underlying DNA sequence. During critical developmental windows, such as embryogenesis and early life, these epigenetic marks are being laid down rapidly and in large numbers. Folate, by donating methyl groups via the one-carbon metabolism cycle, is one of the key architects in this process. Without sufficient folate, the fidelity and precision of this methylation landscape can falter, with consequences that may not manifest until much later in life, or even in future generations. One of the most intriguing aspects of this folate-driven epigenetic machinery is its role in fetal imprinting. During early pregnancy, DNA methylation patterns are established in the developing embryo and are essential for regulating cell differentiation, organ development, and even the establishment of metabolic set points. The maternal folate status at this stage can influence the methylation patterns of genes associated with metabolic regulation, immune tolerance, and neurological development. As a result, folate availability during pregnancy is not just protecting against anatomical malformations, it is also quietly shaping the child's lifelong risk of chronic diseases such as obesity, diabetes, asthma, and even certain cancers. What is even more astonishing is that folate-sensitive epigenetic modifications may extend beyond the direct child and affect subsequent generations, a phenomenon known as transgenerational epigenetic inheritance. For example, studies in animals have shown that folate deficiency in a grandmother can influence DNA methylation and disease risk in her grandchildren through both the maternal and paternal lines. These effects are believed to be mediated through germline modifications, epigenetic marks placed on sperm and egg cells that survive the reset phase during embryonic development and carry forward altered gene expression tendencies. In humans, emerging evidence suggests that paternal folate status prior to conception may affect sperm DNA methylation patterns and influence offspring health, adding a crucial layer of responsibility to male preconception nutrition, which has historically been overlooked. Another less appreciated angle is how folate interacts with environmental exposures. When a person is exposed to environmental toxins such as air pollutants, pesticides, or heavy metals, the body often responds by modifying gene expression through epigenetic mechanisms. Folate can modulate the intensity or resilience of these responses by enhancing or stabilizing methylation patterns. For example, individuals with higher folate intake may be less vulnerable to the harmful epigenetic impacts of arsenic or particulate matter, buffering their DNA from aberrant expression shifts that could trigger inflammation or tumor growth. This buffering capacity makes folate not only a nutritional requirement, but also a molecular shield, offering protection against the growing burden of environmental stressors in modern life. Perhaps one of the most overlooked elements of folate's epigenetic power is its ability to influence aging at the molecular level. With time, our epigenetic marks tend to drift, a process known as epigenetic drift, which can lead to dysregulated gene expression, increased inflammation, and higher susceptibility to disease. Folate's continued role in supporting methylation throughout adulthood may help maintain a more youthful epigenetic profile. Some researchers even speculate that dietary strategies rich in folate could slow the epigenetic clock, a biomarker of biological age based on DNA methylation patterns. If true, this would position folate not just as a preventive agent for early life development, but as a possible tool for promoting healthy aging and extending health span. In the broader context, folate's unseen impact on epigenetics invites a reconsideration of how we approach nutrition, not merely as a means to meet daily requirements, but as a long-term investment in gene expression fidelity, generational health, and environmental resilience. Its influence is deeply embedded in the architecture of our biology, operating at a level most of us never consider, yet its outcomes are visible in everything from childhood vitality to adult disease risk and late-life functionality. Through epigenetics, folate becomes more than a vitamin. It becomes a language through which nutrition communicates with our genes across decades and even generations.